This is our project on homeostatic plasticity in epilepsy. Firstly, we will discuss the role of homeostasis in epilepsy, then we will go over our model, and lastly, we will explain our findings. Being inspired by the Balanced Network project, our goal for this project was to model how an unbalanced network can arise. Therefore, we decided to model epilepsy to investigate a pathological manifestation of an imbalanced network. Epilepsy is a neurological disease characterized by seizures in which neuronal populations show hyperexcitation and hypersynchrony. We decided to generalize empirical findings into the idea that epilepsy is the result of a dysfunctional homeostatic mechanism. We will model epilepsy as a result of Hebbian learning, as it allows for unbounded growth without the presence of a mechanism to counter it. In a healthy network, homeostatic plasticity can adjust the strength between synaptic weights to maintain balance, but without this, Hebbian learning may lead to runaway excitation and potentiation. Simply put, our goal was to model seizure activity in a neural network arising from disrupted homeostatic plasticity mechanisms.
Given the information we gathered, we expect our model to demonstrate a balanced network with stable and consistent firing rates when homeostatic plasticity is functioning, similar to the healthy EEG activity in Figure A. Furthermore, when a current is injected, the network should be resilient to it. In contrast, as demonstrated by the seizure activity in the EEG graph B, we expect that without homeostatic plasticity, Hebbian learning will lead to hyperexcited and hypersynchronous firing as a result of runaway potentiation. We also expect to inject a current to cause more excitation in this network. To realize a model that achieves our desired goals, we need to make multiple assumptions. Firstly, within neuronal populations, the neurons have identical behaviors with no variability. Furthermore, we have fixed parameters such as target firing rates, population sizes, and connectivity probabilities. We also assume that these fixed target rates demonstrate balance. Finally, we generalize epileptic causes as occurring from dysfunctional synaptic scaling, not accounting for various other factors.
We needed to find a model capable of capturing the diverse dynamic mechanisms described in the literature, which motivated our choice of using the Ezekowicz model. This model incorporates more biological plausibility than leaky integrated fire and greater computational efficiency than Hodgkin-Huxley-based models. The adapted model allows for more detailed spiking behaviors and control of rate adaptation. This is essential for our aim of producing a pathological state of hyperexcitation because it allows us to more precisely capture maladaptive dynamics. The code defines equations for total synaptic current as the sum of excitatory, AMPA and NMDA, and inhibitory, GABA currents. Each conductance decays over time. AMPA decays at 5 milliseconds, NMDA at 100, and GABA at 10.
We use synaptic scaling, which updates the weights or the connection strength between neurons as our homeostatic plasticity mechanism. W' here represents the updated synaptic weight, which is calculated as a function of the current synaptic weight and the scaling factor that adjusts the synaptic weight. The scaling factor is calculated based on the target firing rate of the neuron minus the neuron's current activity and is divided by the target rate to reflect how far the neuron's current activity deviates from the target. Furthermore, the function is bound by a sigmoid, which is centered around 1. If the neuron fires more than the target rate, the scaling factor decreases, which reduces the synaptic weights, weakens the synaptic influence, and discourages excessive firing by bringing it back to target. In our model, the synaptic scaling updates the synaptic weights more rapidly than biologically plausible, which allows for more efficient experimenting.
Given the previously discussed modeling choices and our approach, we needed to find the right parameterization. This was quite challenging due to the large parameter space, but in the end we built a network of 5,000 neurons with an excitatory to inhibitory ratio of 4:1, similar to how real neural networks balance activity and what we've seen in the Canvas project. The excitatory neurons exhibit regular spiking behavior, while inhibitory neurons exhibit fast spiking, which is also what is seen in the cortex, especially pyramidal cells and interneurons. Additionally, we included 5,000 randomly firing Poisson neurons. The connection probabilities that you see here are supposed to reflect an unhealthy state of external sprouting, which means that we increased the excitatory to excitatory connections. Reflecting one of our key assumptions that we discussed before, we set the target firing rate for excitatory neurons to 4 Hz and for inhibitory neurons to 14 Hz. This is inspired by experimental data. Additionally, we also injected a small current into the excitatory network so we can study the response of the model under stress. Overall, we chose a combination of biophysically and phenomenologically motivated parameters. As already discussed by Anna, our synaptic scaling is updating very quickly and not biologically plausible. This is evidenced by the very quick oscillations or changes in synaptic weights in a span of 10 seconds. However, we can see that our model effectively compensates for an injected current, which occurs between 2 and 6 seconds. On the right, we assumed an impaired homeostasis to be similar to Hebbian learning, which is characterized by unbounded growth in the excitatory to excitatory connections. But what effect does this have on the assumed healthy average firing rate of the population? To answer that question, we will focus on comparing the average firing rates for homeostatic plasticity on and off. With homeostatic plasticity turned on, we can see that each population is oscillating in the ranges that we set it to. We can see that the inhibitory neurons, or the inhibitory population, is roughly oscillating around the average of 14 Hz, and the excitatory neurons are oscillating roughly around the average of 4 Hz. We can also see that the whole network is responding to the onset and offset of the injected current. Also important is the fact that each population, or that both populations, are interacting with each other, where the trough of the inhibitory neurons is followed closely by a peak in the excitatory neurons. When the homeostasis fails and we basically just have Hebbian learning, we observe none of these characteristics. It shows steady activity above the assumed healthy level with no sign of adaptation. Additionally, it can be seen that the inhibitory population is not able to properly respond and govern the heightened excitation anymore, as there is no interaction between the both populations. Especially in the injected current period, you can see that there is no response to the current from the inhibitory population, which is the case in the homeostatic plasticity on condition. To conclude, it can be said that homeostatic plasticity is crucial for our network to return to a physiological state, especially when axonal sprouting occurs. It is also important for the network to retain responsiveness to stimuli. It also effectively prevents hyperexcitation with otherwise pathological connectivity. With these conclusions, it is however key to remember the simplifications we took. For example, we assume a healthy balance to be evidenced by the average firing rates. In reality, however, pathology manifests itself in electrophysiological measurements like an EEG and not rate measurements. Additionally, the hyperexcitation observed may indicate a seizure. However, we also expected hypersynchrony in form of oscillations, which we did not see, which means that we were unable to confirm part of our hypothesis, of our expectations. This may partly be due to our way to record, so we didn't use an EEG-like measurement. Another previously mentioned challenge is the huge parameterization. The model grew and so did the parameter space, which made it particularly hard to fine-tune the network. So now it is hard to say whether our findings are actually significant and not simply a product of the wrong initialization or parameterization. Then that's it. We thank you for listening and paying attention to our projects, and we hope that you enjoyed it. I suspect that you won't really be interested in the references. However, the next slide also includes a link to the GitHub repository with the code, which you will maybe also not be interested in, but that's fine. And thank you very much.